Hello, I'm Amir and this is my YouTube channel Genetics with Amir. As you can see, the topic of this video is about DNA packaging. Imagine trying to fit miles of thread into a tiny box. That's basically what our cells do with DNA. But how do they fit all that genetic information into such a small space? Let's dive into the amazing world of chromosomes and find out. Every cell in our body has a nucleus, and inside that nucleus is DNA. DNA is like a super long twisted ladder that contains all the instructions for building and running our body. But if you stretch out all the DNA in a single cell, it would be about 2 meters long. That's a lot of information to pack into a tiny nucleus. To fit all that DNA inside, it needs to be super tightly wound. This is where proteins come in. There are two main types, histones and non-histones. Histones. Think of histones as spools. DNA wraps around these spools, forming structures called nucleosomes. Non-histones. Non-histone proteins help to further compact the DNA. Histones play a crucial role in DNA packaging by providing a structural framework for the long DNA molecules to be tightly coiled and organized within the cell nucleus. Here is a breakdown of their key rules. Nucleosome formation. Histone octamer. Histones form a core structure called the histone octamer, consisting of two copies each of four core histone proteins, H2A, H2B, H3, and H4. DNA wrapping. The DNA double helix wraps around this histone octamer approximately 1.65 times, forming a structure called the nucleosome. Also, histones are essential for organizing chromosomes, gene regulation, gene expression, and cell division. Non-histone proteins play a crucial role in DNA packaging alongside histones. They contribute to the organization, compaction, and regulation of chromatin structure. Here are some of their key rules. 1. Structural organization. Scaffold proteins. These proteins provide a framework for the higher order folding of chromatin fibers. 2. Gene regulation. 3. DNA replication and repair. 4. Nuclear organization. Levels of packaging. Once the DNA is wrapped around histones, it coils up even more, forming a thick fiber. This fiber then folds into loops and supercoils, becoming even more compact. The final supercondensed form is what we call a chromosome. Chromosome structure. Chromosomes have a distinct X shape with two identical sister chromatids joined at the centromere. These chromatids contain the same genetic information. Why it's important to know about DNA packaging. Understanding how DNA is packaged is crucial for understanding how genes are turned on and off, how cells divide, and even how diseases like cancer occur. Why is DNA compaction necessary? DNA is incredibly long and needs to be tightly packed to fit within the nucleus. This compaction also helps protect the DNA from damage and facilitates chromosome segregation during cell division. Conclusion. The packaging of DNA into chromosomes is a complex and essential process that enables eukaryotic cells to store and organize their genetic information efficiently. By understanding the mechanisms involved in DNA compaction, we can gain valuable insights into gene regulation, cell division, and human health and disease. Thanks for your support.